The influx of boxes on my Zoom screen seems to have slowed, so perhaps, as I say, that's a small pop-up, perhaps it's time to start. So kia ora tātou, ko Mick Black tōku ingoa. Welcome along. I am Mick. I am the other half of the Peter Mick co-director team for Genomics Aotearoa, and I am on hosting duties today, and it is an absolute pleasure to be able to introduce to you Dr Joseph Guhlin. So Joseph's been with Genomics Aotearoa for how long now, Joseph?
Like 4 years.
Yeah, forever. You pretty much seem like you become a permanent fixture here, and it's fantastic we've got you here. So Joseph's background is in Plant Microbial Sciences from University of Minnesota, but despite the very biological sounding twist to that, he is a programmer, and all of my interactions with Joseph in particular have been very much in that programming space. A broad range of interests: small population genomics, high performance computing, gene family expansion and contractions, applying machine learning to small populations and genetics. Today, Joseph will be talking about his work on the Kākāpō project as part of Genomics Aotearoa high quality genomes program. So I'm going to hand over to you, Joseph, because you've got lots to talk about and I'm taking up time. So thanks for coming along, and really looking forward to hearing what you're going to say. I've lost the ability to speak. That's how excited I am.
All right. Thank you, Mick. Cool. Thanks for the introduction. Good to see everybody here. I know many of you, but not all of you. I hope you find this interesting. It's only my name on the presentation, but I can't stress enough that this was a huge collaboration, and I actually have the complete list of authors at the end. So today we're going to talk about species-wide genomics of Kākāpō, providing transformational tools to accelerate recovery. This is on bioRxiv, but it's also going to be published in Nature, Ecology, and Evolution, and we just got the proofs back today, and we have to get them completed by Monday. So very exciting, and that will be the improved version of the paper. So Kākāpō is Aotearoa's most famous bird, I believe, and they're incredibly adorable. And they like to do close-ups. Now, when we had submitted the paper, they were limited to a few offshore islands, predator-free islands that are managed intensively by the Department of Conservation, although in times past, they have been found all over Aotearoa. I guess one of the best or worst things is to be completely outdated immediately. But as of a few weeks ago, Kākāpō have returned to the mainland. Doc has translocated 4 boys. I believe it was outside Wellington. Shoot. Anyways, but Kākāpō are back on the mainland. It's absolutely historic and wonderful. But previously, all over, now just a few intensively managed isolated populations, predator-free islands with constant monitoring to prevent rats and stoats coming on. And this is really cool from, I mean, this is good for, you know, an endangered viewpoint, but it's also really cool from a data scientist, data nerd viewpoint, because we get a lot of data back that we can play with. And I'm going to introduce you to some of that today. So Kākāpō are a Tonga species. This means they're treasured by the iwi and under their domain. We had sequenced 169 individuals. This included all living individuals at the time. I think there were a few that failed. This is all done with 2nd generation sequencing, so, you know, short reads, Illumina. It is a small, highly inbred population with a limited founding population. And again, it's been a large consortium for this paper, and, you know, even just getting the reference genome out was a huge undertaking with a lot of other people. So, you know, by the 1920s, they were thought to be extinct on the North Island. In 1970s, nobody had seen them at all. In the late 70s, they captured 18, but they were all males, which is not how you start a breeding program. At some point later, they did catch some females, and a recovery program was established in 1995. So sequencing project, second generation, collaboration, Naitahu, Department of Conservation, Genomics Aotearoa, many others, especially ECRs, CRIs, and different universities. And we also were very fortunate to have a really high quality reference genome from the Vertebrate Genomes project. This is from the New York lab with Eric Jarvis and all. And that's that 169 individuals. Back to Zoom, so very large graphs. You can see we've had a severe population bottleneck. So our founding population is about, I believe, 54 individuals. And this is from Wikipedia, so it's a little outdated, but we are now at over 200 individuals with the latest few breeding seasons. So really exciting to see the population growing. This is their pedigree as visualized as a graph in Gephi. And it's not a family tree. And I believe Patrick coined this phrase, but apologies, Patrick, if it wasn't you. It's more of a family bush. And that is the best way I found, best description I've ever heard for this. So there's a significant amount of inbreeding going on, not just from the original founding population, but ongoing, just to try to maintain that variation level in this managed population. So our first step was to do variant calling, and high quality research is absolutely critical to bring this all the way to the application level, which is something we wanted to do from the beginning, not just have a descriptive paper, but something, outputs that can be used for conservation purposes. So we wanted to get really high quality variant calling. To do this, we tested out 3 variant callers. We did freebase, GATK, as well as deep variant. Because we have a pedigree, we're very fortunate that we can look at Mendelian inheritance errors. And when I say this, I don't mean filtering at Mendelian ratios of the population, but actually looking at how many errors are found. So if you have 2 parents and they're both homozygous for cytosine, and the offspring is homozygous for a completely different allele at that site, you've got one of 2 problems. Either the variant caller is messing up, or you've got different parentage than what was recorded. So a few were actually corrected by Marissa, which was absolutely wonderful. But we were able to use this as kind of an error metric to improve on our variant calling and see which ones gave the most errors, which ones performed the best, and get that high quality step. So we actually trained a deep variant model using this data set, using this Mendelian inheritance error. And we did this by subsampling the read level down. So for very high confidence SNPs, so no Mendelian inheritance errors. In fact, Mendelian inheritance agreement, where you have the trios. So kind of the trio binning for doing the assemblies, but this is for SNP calling, subsampling the read depth down, and we were able to get it. There's a little drop after 350,000 epochs, and that was changing the subsampling a little bit to improve for heterozygosity detection. And we're able to compare these all. So deep variant GATK, and then GATK with a SNP quality greater than 250. You'll notice freebase is missing. Its Mendelian inheritance error ratio was incredibly high, and which is unfortunate because I've used it for projects in the past. And also because it took an inordinate amount of time to do the SNP calling. Denny is either laughing or cringing at me. So here on the y-axis, we have the level of Mendelian inheritance errors that we're able to detect in the entire population. And the genotype quality in blue is deep variant, GATK, and then GATK, but filtering for the population level quality greater than 250, whereas GQ is the individual genotype quality SNPs. And you can see deep variant is already outperforming, and they call about the same number of SNPs. So this was really exciting. If you even do a little bit of filtering with genotype quality at 10, you can see that errors really shrink. It's really exciting, and so on. Although if you do filter GATK at higher levels, you can get very close to the deep variant performance. Now, I do want to say again, this is deep variant trained model, not the deep variant default, which is human and E. coli trained. So that's an important takeaway. I don't have those numbers here, but it's something to keep in mind. If we do the genotype quality greater than 50, just looking at the total number of Mendelian inheritance errors, you can see that deep variant is almost at 0, whereas GATK and GATK but filtered is still showing some errors. So that's really cool. We're actually getting a really high quality variant data set for this population. GATK can get close to deep variant accuracy with heavy filtering. You do remove a lot more SNPs though. And even with strict filtering, you get some trio errors, but both methods miss valid SNPs the other find. And I feel confident saying this because I'm the crazy person that will open up IGV and look at several hundred SNPs and the BAM files just to kind of check everything by eye. But we could retrain deep variant. We could take the highest quality GATK ones, treat them as gold standard, and then retrain deep variant. Now, part of this is getting outputs, not just a research paper with a lot of awesome people, but other outputs as well that's going to be useful. And what we have here is a trained deep variant model, which has already been used for a new generation of birds, and it's going to be used for another generation of birds. I believe that sequencing was just sent to me this week. The training takes a lot of time, although doing it on Nessie on the GPUs, I think it was about 2 weeks. Once it's been trained, deep variant runs really, really fast. So that's nice. One-time cost at the beginning and a shorter cost at the end and for future. And that model also works in the newer versions with deep variant. So that's cool. So we have another thing we need to talk about, and that is we have a very small sample size, but we want to get all the way to conservation applications, not just descriptive. And how do we disentangle the threads? It's going to be a really noisy data set. It's going to be a small data set. And to do that, we have to do a lot of simulations on what we can detect. So what levels of heritability can we detect? So this is running heritability. This is running, I believe, the entire Bayes alphabet, although I think maybe 1 or 2 new ones have been added in the last couple months, as well as Tassel, which is just, looks like it was developed in the 90s, but it's a good tool for doing GWAS and a little bit of mangling of SNP files. And we're able to see the absolute error. We ended up using heritability as defined from Tassel as well as Bayes C, because we get the smallest number of errors. This is with several thousand simulations each for a binary phenotypic trait, randomly chosen SNP at different allele frequencies, and that's just kind of bunched in, which gives you the errors. And this way we're able to figure out where we can detect, what we can detect. We're also able to do this by adding the number of causative SNPs. So we can change the number, instead of a single binary trait, we do several causative SNPs for this. And we're able to basically run the same thing. And it actually all looks pretty good, especially if you're looking at Tassel and Bayes C. Oh, I have a little red marker, don't I? Yeah. We've got tassel over here and base C right there. And it doesn't matter how much, whether it's 10 causative SNPs or 1, as we previously saw, or 10,000 causative SNPs, the absolute error is the lowest on these. That's not to say they're the absolute best across the board. For our population structure and for how we did the simulations, these are the ones that are working. But you know, if you're going to do conservation research or really small population stuff, I definitely recommend doing a bunch of simulations. Oop, there we go. The other thing is probabilistic programming. I'm going to go into a little bit of advertising mode here. I'm hoping to run a workshop on it one day, but for all of you who are heavy into stats or heavy into programming, this is a marriage between the two of them. And as a programmer, this gives me superpowers when it comes to stats. So it's really exciting and really great for me. So computer science. Oh, I'm going to be annoying with that red marker now. You've got your parameters, your program, you want to get your output. For probabilistic programming, you're going to start in statistics, you start with your observations, you're going to come up with the methodology, and then you're going to kind of do the parameters, figure it out so you can infer what parameters arise from these observations, or even you can kind of flip it and determine which parameters can give you those sort of observations. There's several different ways to do this. There's STAN, which is kind of the old school way. There's Pyro, which is for PyTorch, and that's from Uber, the app on your phone. And they use that to figure out how much to charge you for surge pricing, and probably some other stuff. There's also TensorFlow. So this is Google TensorFlow probability. It does let you make Bayesian neural nets, but we don't have to go that far for this. There's also Turing.jl if you're into Julia. And I encourage everyone to dig into it. There's a lot of great tutorials out there. So whichever programming language you like, you're good to go. TensorFlow probability also runs on R as well as Python. So should be covered there. So our goals, we want to reduce the environmental noise in the data. We want to figure out how much the environment and other factors are providing, are affecting our phenotypes. Not only to remove it so we can associate phenotype with genotype, but we can provide this information back to the Department of Conservation. Hey, birds on island X growing really fast, island Y growing not so fast, something's going on. So it's kind of a, you know, a holistic data science approach. Not only do we have genotypes, we have phenotypes, and just from the phenotypes, we can get really good information that, while it doesn't satiate the genomics nerd, does help the survivability of the species, and that's really important. But then we can feed these, and you know, we can figure out how much the bird contributes to these, and feed that to breeding value tools, figure out the breeding values. And then that can be used for prioritizing decisions at the Department of Conservation, translocations. There was some illness and they had to prioritize healthcare. So this can be really useful information for the conservation management team. But I said we had a really small sample size, 169. Not all of them have phenotypes. Here we're looking at clutch size. So I'm a plant scientist, but even I know only females lay eggs. And then depending on their age and just chance, how many clutches they've lain, you know, so they may have a single data point, they may have multiple data points. So as you can see, we've got about 40-ish females, and many of them have only a single data point, and some have a few more than that. And clutch size, so they lay between 1 and 4 eggs. That's the important thing. And so, and huge thank you to Anna for helping me convert my Python into math. I'm not going to go deep into this, but it is cool. I'm very proud of it, even though she understands more of it. But we're able to create everything in this model and everything underneath it as a bit of a distribution, and able to do random effects for remove fruit. This is the fruit that ripen when they're going to breed. So they don't breed every year, they breed every couple years. And then whether it's their first or second clutch. So Department of Conservation came out with a really awesome trick. If you take the eggs away after they laid them, they will go and lay more eggs. This is really important for a species suffering like this. So that's really cool. So I'm going to do this more like programmer mode. But we've got our model, we've got a quantized normal distribution where we're taking the intercept plus the effect of the bird, the ripe remove fruit, and the clutch. Observations are tested against Poisson distribution. Do they fit this distribution? So in 2019, they might have 3 eggs. 2016, they have 2. 2010, they have 4 eggs. And this helps us identify variables that make up the distribution. Ooh, there we go. So, and some of you will roll your eyes, but as someone who grew up as a non-Bayesian statistician, this is really amazing to me. Everything's a distribution. So the intercept, the contribution of the bird, the ripe remove fruit, clutch, their distributions. But actually each bird itself is a normal distribution. Each, both of the remove fruit, absent and present, are a normal distribution. And clutch, first or second, are normal distributions. And they get standard deviations, which kind of gives us how confident we are in that, as well as how, what's the natural variation. We're able to define this in Python. So we're able to define a model. We generate an surrogate posterior, is what they're called. And then we're able to train that to reduce the negative log likelihood that it creates a distribution that matches the distribution, the actual observations that we see. So could the actual observations come from that distribution? And so we're able to actually view these clutch size effect estimates, these actual effect estimates. So we've got our intercept. They all seem to want to lay about 3 eggs. The effect of ripe remove versus no ripe remove available. So you can see if there's no ripe remove available, that decreases. It could be a nutritional thing. It could be something else. This information we can feed back. Their first clutch, they generally may lay more eggs. Clutch B, they'll generally lay less eggs, although this distribution can go above as well. And then you can see this is all 48 of the birds. They have a very tiny effect on the clutch size. But we are able to feed this into get breeding values. And you can see starting here, we have the founders, first generation, second and third generation. And one of the cool things that we see is there's a leftward shift of the subsequent generations, but we actually see that as moving back towards the founding distribution, the founders distribution. So that's cool. So some phenotypic, some of the phenotype is lost, some of the phenotypic values are lost, but it seems like it's being recovered with just how the Department of Conservation has been doing their breeding. So that's really exciting. We're able to run this through GWAS, which gives us confidence in our breeding values. And we get a decent QQ plot. That's this plot on the right if you don't do GWAS very often. But we're also getting genes that make sense. And to do this, we search the literature, and especially we found a lot of these GWAS studies that have been done in chicken, although some have been done in ducks, and some of these are GWAS. So the 5th most ranked SNP is associated with egg number in female broilers. We also get differential gene expression studies, egg production in slow and fast growing chickens, abdominal fat, goose egg production. So we find another species. So we're finding the same genes that are being found in other species. Again, really exciting. This gives us confidence in our breeding values, even though it's more of a tiny effect compared to the environmental. It's still really exciting. We're finding genes that other people are finding in other species. And in chicken, especially industrial chicken, you don't have the small sample size problem. And this is the first phenotype that I tackled, which was unfortunate because it was much more complex than I expected. We did growth rate, especially for the first 60 days. So here I want to plot out the effect of hand-reared. So if you're not hand-reared, which are the blue dots, you're going to be on average a bit bigger. Whereas if you are hand-reared, so these are usually a bit sickly chicks, but not necessarily, you're going to be a bit smaller. But as you can see, it's completely overlapping. It's just the general trend. But also sex is really important. So the males grow bigger, the females don't. The females grow a bit smaller. Although you can see some males who were obviously ill, they get removed from the data set. But that's something we can see. So this is again the first 80 days. We cut off our examination at the first 60 days. But you can see that not only do they have some travel in the beginning, but there's areas where their growth slows and actually goes down, and then they're transferred usually to Massey, and then back to Invercargill and Whānau. So they started on an island, got sick, went to university, better, and transferred. But this is all noise in our data set. And so we fit this to a Gompertz curve, and this is back from the day when they hand drew their graphs. This is a beautiful, beautiful example. And so we're fitting here the max body weight as A or M, B, how long to get there, and then the rate of growth. And this is the mathematical formula in Python, actually in TensorFlow Python. And this is why it was more complicated, even though it was the first one I tackled. It's because it's got 3 different parameters for the growth curve, plus there's all the environmental effects. But we were able to do it, and it worked. And we still see that general trend. So we can do the breeding values. Again, founders at the very front. And you can see there's an immediate depression in the phenotypic value, body weight at 60 days. There's complete depression. But you notice the newest generations, they're actually shifting back towards that original distribution. This newest generation has a very wide dispersion. I think that's because of the supplemental feeding. But it's really exciting if you can take an endangered species and then restore it back to the founders' phenotypes and genetic diversity, and then go past that. So it's really exciting what Doc has been doing. Again, we're able to take this, do a GWAS, we get our nice probability plot. It's a little downtrodden at the very top end, but that's all right. It's all about the peaks. Our very first hit, ATRNL1, don't ask me what it does, I'm a programmer. We find it is a GWAS in feed efficiency in chicken and differential expression analysis for abdominal fat in chicken. That's cool. We're finding a gene for body weight that others have found a gene in using 2 different independent methods. Awesome. Feed efficiency in cows, dry matter intake, fat deposition, feed efficiency, body weight gain in broiler chicken. It's exactly ours. Number 81, they actually used a Gompertz curve. So, hey, cool. That's for growth speed. Yeah, so we've got all three phenotypes here. Growth rate coefficient, we've got body weight in Korean chicken, growth rate traits in chicken. There was a disease outbreak. There was aspergillosis. I'm sure you've read about it in the news. This was much harder to do because it was generally younger chicks that got it. They were trying their best to prevent everyone else from getting it. Needless to say, we were able to find a GWAS peak for it on chromosome 1. These are likely aberrations just from SNP density towards the chromosome ends. That's a really cool thing. Whereas this is more in the middle. Cool. So we've got an awesome pedigree. We've got a very high quality variant SNP set. We have life history traits. We have phenotypic traits, which lets us do a lot of really cool stuff with it. Here's the circles plot from our paper. I'm going to just briefly go through it. We've got the chromosome. We have our SNP density. Again, you get SNP density at the chromosome ends. I think it's a bird thing. We look at areas of local population structure outliers. This is local PCA. This is on the green here highlighted is MDS1. We've got MDS2. We're hoping to dig more into these, especially the work coming out of Canterbury with the nanopore assemblies. We're able to look at runs of homozygosity in the founders, as well as runs of homozygosity in the offspring. So that's that inner one. We're able to look at FST between the founders and the offspring, and we're able to look at heterozygosity in founders versus the newest generation. So here, if it's orange and down, the founders have heterozygosity more than is present in the offspring in that area. Whereas in green, we're seeing an increase in heterozygosity as compared to the founders. So we're able to get a really detailed look into the entire population, as well as comparing generations. We've got this local population structure. Definitely want to dig into it more. You've got each chromosome, chromosome 1, chromosome 2, 3, 4, and you're able to see the outliers. If you haven't, I'm seeing a lot more population papers coming out with local population structure. We saw this at ICG a lot. It was getting mentioned. That's really exciting. Doesn't mean I'm doing something crazy and stupid, which is always a giant fear of mine. But we're seeing regions that are really interesting, and both here on MDS1, but also on MDS2. And in the upcoming version, but not on the preprint, unfortunately, we have gene ontology enrichments of these regions, as well as kind of what genes and what regions they are. We can zoom in even more. This is just a region. This is a chromosome. We're able to see transposable elements and genes, but we're able to see putative recombination rates, as well as this local population structure. And we can see that recombination changes that local population structure. This is just a signal finding algorithm. So it means something's going on. And in this case, it's likely recombination. But there are regions where recombination doesn't seem to have an effect. So again, signal finding, and you can see a lot of recombination on those chromosome ends and crazy local population structure. We're able to also take more of a larger view using this. Here, I'm just using local PCA. We've got one outlier bird named Richard Henry. And one of the goals has been to breed him with the rest of the population to kind of keep that variation alive. Here, Richard Henry is highlighted for all the local PCA sections, where he is the furthest out in yellow. So there's some regions where other birds are actually more diverse than he is. And then we're able to look at his offspring and see what these regions are doing, see which regions are more inherited from Richard Henry, which regions are kind of going back towards the general population. So we get these kind of neat little plots. I just wanted to show that. You get here more half and half. But this lets us get a nice detailed view into not only, what variation is being kept, but what variation is not being carried over to the subsequent generations, where it's going. With this high quality SNP variant data set, and this is Stephanie Gala's work, we're able to determine the relatedness of the founders back when the birds were found and then kept. You don't know how related they are or how unrelated they are. And with genomic data, you're able to determine that relatedness and get more of a complete view. This has been used already to determine translocations and artificial inseminations. So it's really exciting. That comes from the high quality SNP sets. Stephanie did a lot of work comparing different methods, and that's in the supplemental information. Again, those simulations, the testing, when you have a small, highly structured population, that's going to bring you so far. So we want to get all the way to applied conservation with this. One of the best ways to do it is this growth rate. One first adorable picture on the right, super adorable. Okay. From this, we have the actual measured growth, and we can see the expected growth. Here on day 17 is when Doc noticed that the bird was not growing as expected. This is not their plots, but this is from our plot, where we're able to take the half value of the parental growth rate, as well as the estimated environmental effects, and we're able to plot the expected growth. And you can actually see at day 6 or 7 is when growth starts trailing off. So one of the ways we hope this begins to affect applied conservation is earlier detection of illness, because if they can be treated even earlier than before, perhaps their final growth at 60 days won't be that much lower. Maybe they'll be healthy. You'll have earlier intervention, better vet treatments, etc. And perhaps some birds who look like they're growing slow are actually because that's what their parents have given them with that half breeding value. Outputs is one of the other big things. So we've got the Aotearoa genomic data repository. We've got our SNP set. If it's not there, it's going to be there soon. It's an output for other people. If people in the future will come in and do much better than me, which will be great. They can use these SNPs, get a head start. They don't have to train deep variant. They don't have to use GATK. It's an output. It helps young researchers, but it also helps the Department of Conservation. Now, of course, you need approval for this. I am not in charge of the approval, so don't email me if it's taking a while. I can tell you who to email. But also we have all of our scripts up on GitHub. So both mine and other contributors. So this goes from QC alignments to the reference, doing a GATK comparison, GWAS pathway enrichment, SNP effect. If you want to see how I created the Circos plot, it's also there. We try to get everything up there. If something's missing, just email me because I'm not deleting any scripts. We've also got all the probabilistic stuff up. As well as my Perl scripts to make Plink work with Camel, work with R, work with Tassel. I did a lot in Perl, but a lot in Python as well. There's also the Bayesian alphabet stuff in R. Not all of my experimentations are there. These are more the final ones, but if you do want to see me failing 1,000 times before I get that nice one, just send me an email. I'm happy to help. As I said, we are up on bioRxiv, but the paper, we just got the proofs, should be coming out soon. Huge shout out, huge thank you to everybody on this list, as well as the Kākāpō recovery team, which includes many, many volunteers, as well as the paid professionals who are recording the weights, clutch size, everything. So, high quality work begets real life applications. It doesn't have to be a descriptive paper, especially in conservation where the need is actually in the applied sector. We can take the genomics work and bring it all the way through. Deep variant, if you're able to use it, it's definitely recommended for humans. I think 20x is equivalent to 30x. Google did a comparison, although, grain of salt, Google's product outperforms someone else, yeah. But we can find appropriate GWAS hits. We can use other literature. We can use studies in other organisms to prove that we're finding good data. These lend credibility to our breeding values, breeding in quotes. We're not going to start breeding them for specific phenotypes. It's just, especially the disease susceptibility, we know, and Department of Conservation makes all these decisions, but who to watch out for, who to give extra attention for, who's more likely to recover. You don't have to help them. Better prioritization of day-to-day on-the-ground activities. And small populations can actually be a strength, not a weakness. All right, thank you.